First of all, I would like to thanks to Florence, Pierluigi, to the whole families here, to the foundation, to give me the opportunity to talk about this unpublished data about models. As Joe Watts has recently talked, the models in these diseases are really important. And I'm going to start with the same animal, the Van Vole, specifically with these particular polymorphisms that, as Joel has described, there are several transgenic models based on these PRP and polymorphisms that are showing very intriguing characteristics, specifically this the degeneration of this spontaneous and infectious disease. This was published by Joel recently, and now, since we have the same animals, we are going to publish also a little more information about the high title that these animals are showing when they are inoculated in voles. But we want to use this information for the starting point to make these questions. Can this phenomenon be mimicked in vitro, this spontaneous misfolding? How would other amino acids, for example, in the same position could affect in the misfolding ability? And also how this position can affect to other species, especially focus on humans. For these reasons, thinking on this particular position and gene, and working in recombinant proteins, we wanted to address what happened when you introduce this important amino acid, but also other amino acids, the rest of the amino acid that we can add. And that's the reason why we construct the whole batch of the different recombinant PRPs, and we test them in vitro. Nature is very intelligent and also gave us the opportunity to show different amino acids in the same positions in different kind of animals, some of them a little bit weird, like bats, kangaroos, elephants, in this position, with this leucine amino acid in this position. But also we have avians with the same, with phenylalanine in the same positions, other little more weird races in sheep also show other kind of amino acid. This is an Asian sheep as well, but also the isoleucine amino acid, the same amino acid involved in this spontaneous generation of infectivity, appears naturally in several species, like Van Voles, of course, but also skunk, or this Mongolian very weird race in horses, a Chinese cow, and also Chinese sheep, etc. Most of the species, as humans, contains methionine in this position, as is described here. So how we are going to evaluate this in vitro, this spontaneous generation or this spontaneous misfolding? We are going to do it using recombinant PMCA, or protein misfolding cyclic amplification, that we have talked several times before. It's a process that occurs in vitro. Basically, I don't want to go to the details. What we want to evaluate is the generation, in red, the spontaneous misfolding in a tube. For that, we use recombinant PRP plus complemented with knockout brain, because we know that when you use the whole brain instead of that is depleted of PRP, you increase the ability for generating this spontaneous misfolding. So what we basically are evaluating in vitro is the number of rounds of PMCA that we need to observe for the first time the spontaneous misfolded recombinant PRP. And this is basically what we are going to show you here. These are the rounds of PMCA that we need to observe for the first time in vitro in spontaneous misfolding. In the wild-type mouse with the leucine in the 108 position, we found the first spontaneous misfolding in the round 8 with isoleucine, as it is the most critical amino acid, appears a little bit earlier. And the third one that appear with valine in a very similar way than appear in the wild type case. The rest of the amino acid didn't show any spontaneous disease, even up to round 20, indicating that just these two amino acids, including valines, are the most efficient to prompt the spontaneous misfolding. So these experiments were performed under rigorous controls of prion contamination. That is a very important issue in this kind of in vitro experiments. But the most important is that these experiments, all of them were done using the same concentration, protein preparation, and PMCA settings. And this is important because the results are indicating that these results are really stochastic. Stochastic means that we observe really differences intra-experiments, so different results using the same protein, everything the same. In different tubes, we have different results. Inter-experiments means that when you use the same samples in the same condition, doing in different days, you have also different results. Inter-protein batches and inter-sonicators, indicating that this process, as probably is happening in humans, is really stochastic. Probably is controlled by something that we don't know now. How we observe these prions in vitro? This is the way that we observe after PK digestions. They are indistinguishable, of course, because they are just different in one amino acid. And we want to address what happened with this protein if it's able to propagate in vitro in the rest of the substrate. And we tested the 20 different substrates with containing different amino acids in the same position, and we observed that not all of the substrates were able to propagate this misfolding. The wild type were very efficient, of course the same substrate that were created, and also other that like these 6 substrates were able to propagate the misfolding. So this is the spontaneous misfolding. Recombinant PAP were propagated efficiently in certain permissive but not all polymorphisms. We wanted to go further, and we wanted to understand if this phenomenon can also be extrapolated to other species. And for this reason, using recombinant PAP, creating this batch of different recombinant from different species. In the top of the slide are those that were able to show also spontaneous generation of prions. Hamster, rabbits, rabbits were suggested to be resistant to prion diseases. We have also horses and of course humans. Some of these experiments are still ongoing. For example, recently we have observed that also pigs can generate spontaneous generation. Some of these species, like dogs, are considered completely resistant, and the in vitro results also match very well with these in vivo results. What we are showing here is 2 independent experiments with each species that were positive, showing in which round of PMCA, of in vitro PMCA, appeared the first case of spontaneous misfolding. As you can observe, different completely independent experiments are showing different results, indicating again that this process is a stochastic process. For example, rabbits appears just in one experiment, but in the second experiment, completely independent, didn't appear up to 24 rounds. And for example, humans was repeated in this case. I'm showing just 2 experiments, but some of these experiments have been repeated much more times. But this is indicated that some species, the stochasticity is not so big. This is the way that we observe the spontaneous prions in vitro after PK digestions. And in some cases we observe, for example, in humans, that we have 2 different kind of strains. From same species, same protein, different experiments showing different, biochemically different strains. With this information, we wanted to go to the animals. This is something, because I have started talking about models. And this is what we basically, the reason why we did these experiments in vitro, in order to choose the right models for understanding this spontaneous diseases, spontaneous phenomenon. We created 3 different lines of transgenic mice based on these particular polymorphisms in mouse. The same that we use in voles, that they correlate with the 109, but this, the sequence in mouse is 108. The first line, overexpressing from 5 to 6x, the founder died in around, showing neurological disease in around 140 days. The second line with 4x show both genders show the same spontaneous neurological disease with a very big window from 150 to 250. And the third line with a little lower amount of PRP, we have the same disease with in around 300 days. When we double the amount of this, the level of expression of these animals just by crossing between them and making homozygous animals, we reduce with a very good correlation with the level of expression that will reduce the incubation time of the disease. So when you have animals and they are showing spontaneous disease, the first thing that you want to know is what kind of disease you have in these animals. And what we have observed in a similar way that the transgenic voles, the animals expressing voles PRP, we have 2 different animals, both showing the same clinical signs, but with 2 different clinical pathology. Mild spongiosis, gliosis, and extracellular PRP deposition accumulations is a standard TSC. In another cases, we have just perineural staining, not so very clear standard TSC. But both of them show the typical or the atypical prion band. This is after PK digestion, similar as Joel has shown before. So we have recreated in a different PRP, in different species, using the same polymorphin, a very similar disease. So the next question was what happened with infectivity? Because if this is happening in vole, and now we have mouse, the most important point is to go to humans, and this is basically what we are doing now. But first, we wanted to clarify if these animals are sick animals, were also infectious. And we use TJ20, it's a very well-known model, and using 2 different experiments, using as substrate, as inoculum, 2 different lines from brains from sick animals, and in both cases we observe a very clear disease, a prion disease in a very short period of time, showing the same atypical band that I have talked before. So now we are finishing the experiments using the wild type animals as a model, and we are observing now most of the animals showing clinical signs, and now we are close to 300 days, indicating that as a transgenic overexpressing voles PRP, these animals expressing the same the same protein, but in mouse species, are showing the same characteristics. So the conclusion for this study is that the position 108 determines the misfolding provenance in most of the species. Some of them even consider resistant to prion infection. Not all the amino acid changes support the spontaneous misfolding, only isoleucine, leucine, and valine. A spontaneous misfolded 108 mouse recombinant PRP was propagated efficiently in certain permissive but not all polymorphisms. And the substitution of isoleucine increased the misfolding tendency in at least 10 different species, including humans. The 3 transgenic mouse lines generated with relatively low levels of expression developed a fast neurodegenerative disease that was transmitted to wild-type mouse model very fast and showed an atypical prion disease. So now we are generating different models based on the expression of human, sheep, and cow in order to recreate these diseases in different species. Of course, I want to finish acknowledge to my people in the lab, but also many other groups, and of course, the foundation for giving me the opportunity to be here. Thank you very much.